All right, so we'll pick up here by talking about the spinal cord. And so our spinal cord is the other part of our central nervous system. When we discuss the embryonic development of our uh, brain and of our central nervous system, we talked about the neural tube, we talked about a neural plate folding back into a neural groove and ultimately forming the neural tube. And the very end portion, the very caudal portion of this is going to be our spinal cord. Now, remember that it is derived from ectoderm, just as our skin is. And our spinal cord is going to send and conduct different nerve impulses. So, impulses will travel from the brain down the spinal cord out to our tissues. And then it can also transmit signals from the tissues to the spinal cord and up to the brain. So, it can transmit motor signals or efferent signals as well as afferent or sensory signals as well. It can also coordinate spinal reflexes, so things that occur involuntarily and autonomically, and we'll talk about some of those in a little bit. So, our spinal cord is the kind of distal extension of the brainstem as it enters and passes through the foramen magnum as it leaves the skull. It travels down through our vertebral canal, through the vertebral foramen of the different vertebrae, and it ends about the region of our first lumbar vertebrae, or L1. Here it becomes something known as the conus medullaris. There is a little string-like structure, this piece of connective tissue known as the filum terminale, that will extend down and anchor the spinal cord down to the coccyx, and we'll look at some pictures. And then we have something known as denticulate ligaments, and these are little regions of pia mater. If you remember pia mater, is the innermost or deepest most meningeal layer. And these denticulate ligaments are these little extensions of pia mater that attach to the spinal cord and hold it at least somewhat in place. Now, at the region where our spinal cord terminates around L1, the first lumbar vertebrae, it's going to change name from just the spinal cord, which is just this fibrous cord, to something known as the cauda equina. And what cauda equina, cauda refers to tail, and equine or equina refers to horse. So this is the horse's tail, and what it is it's this cluster and this aggregation of nerves, kind of at the terminal end of the spinal cord, that extend down still through the vertebrae, and then will extend out to the body and provide it with different types of innervation. Okay, so we'll take a look here. Here you can see the end portion of your spinal cord here. So if this is L5, 4, 3, 2, 1, it's ending right about here at L1, and then you get this formation of the cauda equina, which is this horse's tail appearance of these nerves here. And you can see way down here, right about where my cursor is, you have a single little connective tissue extension here, and this is going to be your filum terminale that will attach and kind of anchor your spinal cord down here to your coccyx. Now, at different regions on our spinal cord, we have some enlargements or some areas that are kind of a little enlarged and swollen. We have a cervical enlargement and we have a lumbar enlargement. What these are for is we have major nerve plexuses that leave our spinal cord and go out and supply our extremities. So we have one large one here in the cervical, the lower cervical and upper thoracic regions known as the brachial plexus, supplying innervation and nerve supply to our upper limb and muscles and cutaneous innervation there, so sensory innervation. And then we have one down at the lumbar and sacral regions known as the lumbar plexus and sacral plexus, and sometimes they're referred to collectively as the lumbosacral plexus. Okay, so these are these different regions that are enlarged for this region where we have nerves exiting. When we look down at our spinal cord from a transverse section, we can see different areas. We can see some regions that have this kind of invaginated appearance. So we have something known as an anterior median fissure and a posterior median sulcus. A fissure is, if you remember from one of our previous parts of this lecture, a fissure is a deep groove and a sulcus is more of a shallow groove. So here you can see on your anterior median fissure, it dives way deep down, whereas our posterior median sulcus, there's just a smaller, more shallow groove. And we can also see some white matter and some gray matter regions here that we'll talk a little bit more about in a second. So as we enter or leave our spinal cord, we do this through different roots. Okay, so we have a dorsal or a posterior root, and we have a ventral or an anterior root. The dorsal root is going to be where our sensory or afferent neurons are coming into the central nervous system from the periphery, so from some peripheral structure. And so what you'll see is that it will enter into the posterior aspect of our spinal cord via this posterior root or dorsal root. And then you have a kind of opposite ventral or anterior root, which is exiting through the ventral surface of the spinal cord. And this will contain our motor neurons that are then leaving the central nervous system, leaving the spinal cord, and going out to the periphery to carry some type of motor signal. Okay, so if we look here we can see a dorsal root, so we know that signals are going this way from the dorsal root into the spinal cord, and here we see a ventral root as signals are leaving the spinal cord, entering out, and they will join the spinal nerve and go out to our body. Now, if we take a closer look at our dorsal root specifically, you see this kind of swelling. So if I go back here, you can see this swelling right here. You can see it on the opposite side as well. And then here you can see a large swelling here, and you can see another one right here. What this swelling is something known as the dorsal root ganglion. And so in a previous part of this lecture, we referred to ganglion and nucleus. Nucleus was a collection of cell bodies within the peripheral nervous system, and a ganglion was a collection of cell bodies outside of the central nervous system. And so if you know, we're not in the brain here, we're not in the spinal cord, we're out here to the side. And so this is a collection of sensory neuron cell bodies that is outside the central nervous system. So we're going to call it a dorsal root ganglion because it's a part of this dorsal root. Okay, and again, this contains purely cell bodies of sensory neurons. Okay, now you may be asking, if this is the cell body, if these are the collections of cell bodies of sensory neurons, where are the cell bodies of the motor neurons? Well, those are going to be located here in this region that we'll call the ventral horn or anterior horn of gray matter. So when we look here now at these horns, like I said, we have a dorsal horn and we have a ventral horn. The dorsal horn is where we will synapse with sensory neurons as these interneurons continue forward to synapse with our motor neurons. The ventral horn, as we just mentioned, which we'll see right here, is going to contain the motor neuron cell bodies. And then we have, in just certain regions, something known as the lateral horn. And in this image here, you can't really see it. You can see it a little bit as this kind of region. Actually, you can. This is not going to be the lateral horn because we're in a cervical vertebrae here, so part of the cervical spinal cord, and you do not contain lateral horns in the cervical spinal cord. But what it is it's going to contain the cell bodies of sympathetic autonomic neurons. Okay, so part of our autonomic nervous system, and these are going to be only located in the thoracic and upper lumbar region. So specifically about T1 all the way down to about L2 is where you'll have sympathetic outflow. And when we discuss our sympathetic nervous system and autonomic nervous system, we'll go into more specifics on their function and kind of the layout of their neurons. So what we see is we see our dorsal horn, we see our ventral horn, and here we can see our lateral horn. So again, if we see our lateral horn, we know that we're somewhere between T1 to L2, somewhere in the thoracic region or the very upper parts of our lumbar region. Okay, and again, when you see these, start to associate what's going on. So lateral horn, cell bodies of sympathetic neurons, ventral horn, cell bodies of motor neurons, and when we see our dorsal root ganglion here and here, this is the cell bodies of sensory neurons. Here we can see the same thing. We see our lateral horn, we see a ventral horn or anterior horn, and then a posterior horn. We also have a central canal. This central canal is another route where cerebral spinal fluid will be able to flow. And we'll then talk about these different areas of white matter that kind of surround this ventral and dorsal horn of gray matter. Here, if you follow into your spinal nerve, you can see the dorsal root ganglion. You can see a cell body of your sensory neuron. It will enter into this dorsal root. It will continue as this, or synapse with this interneuron connecting the sensory and the motor neuron. You see the cell bodies of your motor neuron here in your ventral horn of gray matter. It will enter out through this ventral root and then out your spinal nerve and out. Okay, so make sure you keep in mind the difference between the dorsal and ventral root and the dorsal and ventral horn. They're different. The ventral and dorsal root are these nerve roots that are entering into your spinal cord. The dorsal and ventral horns are the collections of gray matter within the spinal cord itself. Okay. So one of the last things we'll talk about here is our something known as white columns. And so we have a couple of different columns of white matter. And so if we're dealing with white matter, we know this is going to be axons of neurons that are myelinated. Okay. We have 2 anterior, 2 lateral, and 2 posterior. Now, these columns will contain subdivisions, or you can divide them into these more specific parts known as fasciculi or a fasciculus. Okay, what these are they're bundles of nerve tracts within these white columns, so they contain the myelin, that have a specific destination. Okay, or they have a specific function, and these are going to either be ascending or descending. Ascending are going to be those that are sensory or afferent, that are coming from the periphery, ascending in the spinal cord and heading up toward the brain. Descending are going to be motor or efferent tracts that are going to be coming from the brain down the spinal cord, out a peripheral nerve, into some effector tissue, maybe like a muscle. Okay, so again, ascending is sensory signals, descending is going to be motor signals. So when we look here, we can see these different columns. So here you can see your posterior columns, here you see lateral columns, here you see anterior columns, and you can divide, and you have 2 of each, and you can divide those even further into individual fasciculi, and some will be ascending and some will be descending. So in this image, everything in blue is going to be ascending, everything in red is going to be descending. So if you see something in red or descending, we know that that is a motor signal that is going out to a very specific part of the body and carrying a specific signal. If you see it as blue, it is ascending. You know it is carrying some type of sensory stimulus. So whether it's proprioception, whether it's touch, some type of pressure, vibration, pain, anything like pain, temperature, all of these will ascend in these different ascending white column tracts that are going from the periphery to the spinal cord and then ascending up to the brain. Okay, so we'll talk here just briefly about some spinal cord disorders, and then we will wrap up. So there are some different terms to be familiar with. If you hear the word paralysis, this is a loss of motor function. If you hear the word paresthesia, this is loss of sensory function. So a loss of motor function would prevent you from moving. It would prevent some type or some degree of movement. We know that people can be a hemiplegic or a paraplegic or quadriplegic. So quadriplegic meaning you have lost motor function in all 4 limbs. Paraplegic meaning you've lost it in about 2. Sometimes hemiplegia can refer to 1/2 of the body. And then a paresthesia, you lose sensory function. Okay, so you may see in a clinical setting, patients may have paresthesias. They get this loss of sensory function, so they may have tingliness or numbness in their extremities, in their periphery. And so there are different medications that can combat paresthesias. You have something known as poliomyelitis or polio. This is destruction of ventral horn neurons by the polio virus. And so think about it, if you're destroying the ventral horn, or the neurons or the cell bodies that are in the ventral horn, also known as the anterior horn, what are you destroying? Well, you're destroying the motor neuron cell bodies. So if you're destroying the cell bodies of motor neurons, you're going to have difficulty initiating and having control over different types of motor function. Okay, and then you have this last one we'll talk about, ALS or amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, and this is a progressive disorder of, again, your ventral horn motor neurons, those cell bodies, and so this is a very progressive motor disease that slowly eats away at an individual's motor function. Okay, so I guess we actually have one more slide here, and then we'll be done. So yeah, so we have some diagnostic tests that can be related to the central nervous system or to the nervous system in general. You have something known as a pneumoencephalography. This is something that can be used to assess for something known as hydrocephalus. When a baby is born, sometimes there can be an excessive amount of fluid that accumulates on the brain. So hydro referring to fluid or water, and cephalus referring to the head or brain. And so you'll see these babies, it's actually quite sad. They have this large, swollen head that is just full of fluid. Okay, you can have something known as a cerebral angiogram. This is where you will inject a dye or a material into the bloodstream. It will circulate through the cerebral arteries, and this is something that you can do to determine the patency or the openness of different vessels in the brain to determine maybe the degree of atherosclerosis or determine a stroke in a patient if it is completely occluded. You can have things like a CT and an MRI to be able to visualize tumors, plaques, dead brain tissue. Multiple sclerosis is a demyelinating disorder in the brain that can disrupt motor function and motor control. And then you can have a PET scan or a positron emission tomography in which you have positrons emitted, which are these signals that can be picked up by imaging modalities. You can generally have PET scans include being able to detect if somebody has a tumor. And so what you do is you attach a glucose tracer to this positron. It will the glucose will be attracted to areas of the body that are highly metabolically active. So cancers and tumors are highly metabolically active. And what you have is when it is attracted to this highly metabolically active site or tumor, the positrons will be emitted from that area that's highly metabolically active, and it can be captured by the image. And what you'll end up seeing is something that is it typically is kind of bright orange or red or blue. And this can indicate a particular area of the brain that may have a lesion. And sometimes these lesions can be causing seizures. So it's a good way to diagnose lesions that may be generating seizures. Okay, so we will end there. As always, if you have any questions at all, please feel free to email me, reach out to me. I'll be happy to answer anything to the best of my ability. So good luck studying, and I will see you in the next video.